Now loop of Henle. For example, from here we are taking the Bowman's capsule, and for example this short area is showing the proximal convoluted tubules, and in proximal convoluted tubules we have cuboidal epithelium having brush borders inside lumen. So for example these are the proximal convoluted tubules, and these are the tight. They are called tight junction. They are not called the gap junction. They are called tight junction. काल ले पे उधर गले निकाल सरासरी रे माइक के साथ. So this is the Bowman's capsule. This is the proximal convoluted tubules, and this thing is, for example, this is interlabular artery, and this is the afferent arteriole making glomerulus, and this is the efferent arteriole. Which things are filtering from here? Sodium, potassium, magnesium, calcium, chloride and bicarbonate ions. Glucose, amino acids, lipids and vitamins, small proteins and water. Right? After passing all these things, when passing from proximal, this is the proximal convoluted tubule. When they are passing from the proximal convoluted tubules, for example, this is the peritubular capillary. Right? Sodium is reabsorbing from this area. By which mechanism we have discussed in proximal convoluted tubule? How many? 65%. Right? Water is reabsorbing from proximal convoluted tubule. How many? 65%. These two are very important. Except these, glucose, lactic acids, amino acids, etc., they all are reabsorbing 100%. Right? Now the remaining things, the sodium and water is leaving here. But one thing about the osmolality, what will be the osmolality in proximal convoluted tubule? Here the osmolality is 300 milliosmol per liter. In Bowman's capsule, because all the nutrients, all the solutes are filtering, so the osmolality will be same. Here in proximal convoluted tubules, the same amount of solutes and the same amount of water is reabsorbing. So the osmolality here will be 300 milliosmol per liter. Right? Here, now the next part is the loop of Henle. But for example, generally when this is nephron, this is proximal convoluted tubules. Here we have the descending limb of loop of Henle, and here we have ascending limb, but ascending limb have two parts. One is the thin part of the ascending limb and one is the thick part of ascending limb. Here for example, this is the loop of Henle, or this loop of Henle here, it is narrow. This is loop of Henle. In this part of loop of Henle, we have a very special transporters, and they are always open. Mean in any condition of physiology, in any physiological condition, it will remain open. It will remain open for water. So these are the name of these transporter are, anyone? The name of this transporter is aquaporine 1. Aquaporine 1 transporter, right? And this part is called the descending limb of loop of Henle, right? And this part, what are the specialties or what are the characteristics of this part? This tube is highly permeable to water. Highly permeable to water. And this is impermeable to solutes. Right, impermeable to solutes. What will be the osmolarity? Osmolarity will be, we are discussing. So for example, from here the reabsorption of water take place. For example, this is the peritubular capillaries composed of simple squamous epithelium, right? And from here only the water will leave into the interstitial fluids by aquaporine 1 transporter. This is called aquaporine 1 transporter. Aquaporine 2, aquaporine 3, aquaporine 4, they are present in the distal convoluted tubule, right? But in the descending limb of the loop of Henle, we have aquaporine 1 transporter. Aquaporine 1 transporter, right? And what is the role of aquaporine 1 transporter? It is only permeable to water. It is only permeable to water. If you take an MCQ in your paper, that which part is only permeable to water, not to solutes? The proximal convoluted tubule, the descending limb of loop of Henle, ascending limb of loop of Henle, and distal convoluted tubule. You will select the descending limb because this area is only permeable to water, and this is very important. If the solutes are leaving this area, there will be a problem. So I have told you that when the solute concentration is increasing, osmolality will be increasing. And for example, this is the area which is dipping in the medulla. Here the osmolality is equal to 300 milliosmol per liter. Here for example it is 500 milliosmol per liter. Here it is for example 700 milliosmol per liter. Here it is for example 900 milliosmol per liter, and here it is 1200 milliosmol per liter. Here in this, water is leaving the lumen of the tubule. It means that the solute concentration here become increasing because solutes are not leaving the lumen. It means that the osmolarity inside the lumen is now increasing and it is going up to hypertonic condition, right? For example, here it is 300 millismol per liter. For example, here water is leaving, it is for example 500, it is 500 millismol per liter. Here for example it is 700 millismol per liter. Here it is for example 900 millismol per liter, and here it is for example 1200 millismol per liter. It's mean that water is leaving the lumen and the inside solution is becoming hypertonic, and around this part of the loop of Henle we have 1200 millismol per liter osmolality around this loop of Henle. So it become 1200 millismol per liter. What is the purpose of this 1200 millismol per liter osmolality? It allowing the sodium now to leave this lumen, because in concentration gradient, for example, if this is the cell and this is the interstitial fluids, and here the water concentration is high and the NaCl concentration is low. Here for example, due to increasing osmolality, the concentration of NaCl is increasing and water is leaving this lumen, so the concentration of water is becoming decreasing. It's mean that it form the concentration gradient in these two compartments, and this sodium will easily can leave this cell and can came into the interstitial fluids. So now we will discuss this area of loop of Henle, which is the ascending limb. For example, this is the ascending limb of loop of the first part of loop of Henle is narrow part. Here in ascending limb, this is the narrow limb or narrow part of the ascending limb, and this is the thick part of the ascending limb of loop of Henle, and this is for example this distal convoluted tubule, and this first part is called the early distal convoluted tubules. It have some relation with the loop of Henle ascending limb. So this is the descending limb and this is the ascending limb. In ascending limb we have two parts. One is this one, this is called the narrow part of ascending limb of loop of Henle, and this part is called thick part of ascending limb of loop of Henle. Right here, the osmolality of the solution remaining here is 1200 millismol per liter. It means here the solution is hypertonic solution. Hypertonic solution, because water is leaving continuously, water is leaving this environment and the concentration of the solutes is increasing. Here, this is the thick part of loop of Henle, and this is the cell of right. These are the cells of loop of Henle ascending limb. Right. Of course, the cells will be present on both sides. This is the distal convoluted tubules. Right. We have a very special transporter on these cells, because in this hyperosmotic solution we have a high level of sodium, we have high level of chlorides, we also have a high level of magnesium, calcium and potassium. Right. Here we have a very special transporter very special transporter. Right, and this transporter is called sodium potassium 2 chloride co-transporter, right? What is the name of this transporter? This transporter is called sodium potassium 2 chloride co-transporter. What is the role of this? We have a very hyperosmotic solution here coming from the descending loop of Henle, and here we have sodium, we have chloride, and we have potassium, also we have calcium, we have magnesium, etc. Right? But the very special characteristic, like this is the descending limb which is highly permeable to water and it is impermeable to solute. This ascending limb, this is the ascending limb of loop of Henle. It is highly permeable to solutes and it is impermeable to water. It is impermeable to water, right? It's mean that water cannot leave this area easily, right? Or there is no reabsorption of water occur in ascending limb of loop of Henle, and there is a lot of solutes will leave this area, and this is very important because the loop diuretics, furosemide, Lasix, the loop diuretics acting on these proteins, right? What is the role of this protein? For example, it have three areas. Here. To this black marker, for example, this is the sodium, this is the potassium, and this is the chloride. When all these sites are filled, this protein flipped. It take a turn, right? For example, this is here potassium is attached, here sodium is attached, and here chloride. By filling all these sites, this protein flipped, and when this flipped, the sodium, the potassium, and chloride goes inside the cell, and of course here due to the sodium potassium ATPases, this cell is deficient of sodium, and also potassium is coming to this cell, right? So from this environment, it will take sodium, it will take potassium, and also it will take chlorides from here. How many chloride? 2 chlorides, right? These are, sodium is going inside the cell, potassium is going inside the cell, and chloride is also going inside the cell. We have sodium, we have potassium, and we have 2 chloride. Now here we have, for example, this is that cell. We have 3 special transporter in the cell, and extrinsic proteins, right? They are, for example, one is this red transporter, this red channel, one is the green channel, and one is the black channel, this one, right? What is the name of this? Sodium transporter, potassium transporter, and chloride transporter. It will take sodium from the cell into interstitial fluids. It will take, this one will take the potassium from the cell into interstitial space, and it will take the chloride from the cell into interstitial space. Now look and listen carefully. From here, water is coming into interstitial fluids, and from here, the solutes are coming from the ascending limb. So, when the solutes are coming from here, the osmolality of this area will be decreasing. Why? Because it is taking a lot of water from here, and when it going down, the water is leaving this area, and when we are going upward, the solutes are coming from this lumen. So the osmolality will be remain same, because the same concentration of the sodium, the same concentration of chloride and water will reabsorbing or leaving the lumen of the tubules into interstitial fluids. So when we coming from up to down position, the osmolarity is 300, 500, 700, 900 and 1200. But when we are going upward, solutes are leaving this area, and the osmolarity, for example, here the osmolarity is 1200 milliosmol per liter. Here, for example, it is 900 milliosmol per liter, because the solutes are leaving this area. Here, for example, it is 500 milliosmol per liter, and here it is, for example, 300 milliosmol per liter. But when these solutes are coming to distal convoluted tubule, in distal convoluted tubule, the osmolality is about 200 milliosmol per liter. It means that, and we will discuss in distal convoluted tubules, that this area is hypotonic. This is hypotonic, and this area is hypertonic, and this area is isotonic. Right? This is isotonic, this is hypertonic, this is hypotonic. Right? So this is the mechanism. Here also sodium is leaving, potassium is also leaving, and chloride ion is also leaving. I have a very special question for you people. Potassium is coming to cell from here. Potassium is also coming from here, right? By sodium potassium ATPase. What will be the effect of this potassium? There will be some limited capacities of the cell, but we are looking that potassium is increasing inside the cell. Potassium is increasing inside the cell. It have some effects, right? What is the effect? There are some transporters by which this potassium again leaves the cell and came into this lumen, also to this areas, also to these areas. By coming to this area, all the lumen is getting positive charge, right? Positive charge, and it is becoming, for example, approximately it become plus 8 millivolt. The voltage is plus 8 millivolts. It is due to the potassium present in this area. And for example, here we have calcium, we have magnesium also. They are coming in this hypertonic solution. So for example, I am erasing this area for just a second. For example, this is the wall of, and these are the cells, right? Potassium is like that, sodium is leaving, and potassium is coming inside, and potassium is also coming from here by sodium potassium to chloride co-transporter. The concentration of potassium is high in cell, so there are some special transporters by which this potassium leaves. They are called potassium transporters, right? And due to this potassium, this area is becoming more positive, Now come to physics. What is the effect of same charges on each other? Repelling. So for example, here is positively charged and here is also a very high positive charge, and when this positive charge came here, it will repelled. Same mechanisms occur here. Here is the charges are plus 8 millivolts, and calcium, for example, this is the calcium, and calcium have positively charged. It is positively charged. We have magnesium, and magnesium also having positively charged. When they are coming, they are happily coming in this hypertonic solution, but when they reach here, here is this, the charges are positive, which is plus 8 millivolt. These charges repel the magnesium and calcium, and they are transported paracellularly to the interstitial fluids. So if you have some question that how the calcium and magnesium is transported to interstitial fluids, it is due to the same repelling charges which are provided by potassium to the lumen. It repel the calcium and magnesium in paracellular spaces to the interstitial spaces, right? And this transport is called paracellular transport. This is the whole story about the loop of Henle. But for example, here also this is common. Here will be the blood, and the blood will take these solutes, right? What is the specific name for this mechanism? That from one side water is maintaining the osmolality of this area, and from other side the chlorides are maintaining this osmolality. Or from one side this is impermeable to solutes, and from other side it is impermeable to water. This transport mechanism is called counter current multiplier mechanism. This mechanism is called counter current multiplier mechanism, and this is the story of loop of Henle. What is the role of the vasa recta? So for example, we are taking this is for example, I have nephron, and these are the proximal convoluted tubules, and from here we have loop of Henle, and here we have vasa recta. This is for example vasa recta, and we are taking a lot of sodium, a lot of salts and nutrients, right? So here it takes, for example, from here sodium, from here water is coming. This is the descending limb of loop of Henle. These water are taken by vasa recta, and it gives these water to interstitial fluids. From here, for example, from here it is taking sodium chlorides to this blood, right? And it giving the NaCl to this area. The blood flow in the vasa recta is very slow, and it is used for maintaining the gradient of sodium. What is the role of the vasa recta? Vasa recta prevent the abrupt leaving of sodium from kidneys, right? So this type of mechanism, because it taking the water from outside, right, and here it giving the water, this exchange is called counter current exchange, and the vasa recta is called counter current exchanger. It is called counter current exchanger. This is basically vasa recta, right? And osmolarities are the same. For example, here this is the vasa recta. Here the osmolality is, for example, this is 300, here it is 500, 700, 900 and 1200, right? So this is all about the loop of Henle, right? What is the speciality? Ascending limb is impermeable to water and descending limb is impermeable to solutes. And what is the special transport here? The sodium potassium 2 chloride co-transporter. What is the role, what is the importance of, what is the clinical importance of this transporter? For example, here is the transporter. For example, this is the loop of Henle, and we have the potassium, the sodium potassium 2 chloride co-transporter here. And I have told you that, for example, it is taking sodium, it takes potassium, and it takes 2 chlorides and flips and takes these solutes into the cell and decreasing the solute concentration in the lumen. When we give loop diuretics, these are the medications which are used for high blood pressure, hypertension. One example is furosemide, available with the name Lasix, right? When, for example, this is the loop diuretic, and this loop diuretic inhibits this transporter, and when this transporter is inhibited, the chloride and sodium cannot leave the lumen and the urine amount is increasing. So this is the role of the sodium potassium 2 chloride co-transporter, that when the loop diuretic is attached to this transporter, it is inhibited, and now the sodium, chloride and potassium are not able to go into the interstitium from the lumen of the tubes, right? And after this we will discuss distal convoluted tubule. The distal convoluted tubules are divided into two parts: the early distal convoluted tubules and late distal convoluted tubules. The mechanisms are different in these areas because parathyroid hormone. What is the role of parathyroid hormone in proximal convoluted tubule? It prevents the phosphate to go into blood. And what will be the role of parathyroid hormone in the distal convoluted tubule? It will reabsorb the calcium from distal convoluted tubule. So parathyroid hormones have two functions. One is calcium reabsorption, the second is the inhibition of phosphate into blood, right? So this is all about the loop of Henle. Any question? The side effect da razi kana the loop diuretics इसका urine output पड़े ज़्यादा सेवाशी और जिकल urine output सा सेवाशी इसका पर body के जिकम द गदा मकदार दे द सोडियम क्लोराइड और द कम शी